Hello, everyone. Thank you very much for the great panel that we had and all the great session about sex differences in autism from mouse model to human. My name is Idan Menashe, and I'm the scientific director of the National Autism Research Center of Israel. Before I will present our speaker, I want to mention that there will be time for short question and answer after the lecture. So if you want to have if you have questions for the speaker, please post them on the chat on the window of the public chat on your hand side. And now I'm extremely excited to introduce our keynote speaker for today, Professor Simon Baron-Cohen. Simon is a professor in the Department of Psychology and Psychiatry and the director of the Autism Research Center at the University of Cambridge. He is also a fellow of the British Psychological Society, the British Academy, the Academy of Medical Sciences, and the American Psychological Association. He is currently the vice president of the National Autistic Society and also served as the president of the International Society of Autism Research, INSAR, between 2017 to 2019. I believe that everyone here have heard about the significant contribution of Professor Simon Baron-Cohen to autism research. He is particularly known for his pioneering research and innovative theories about the mind blindness in autism, the prenatal sex theory, the emphasizing systemizing theory of typical sex differences, which is also known as the extreme male brain theory. He is author of over 600 peer-reviewed scientific articles and 5 books. Professor Baron-Cohen is also involved in many public activity for people with autism. In 1999, he created a class clinic for adults with suspected Asperger's syndrome that has helped over 1,000 patients that have their disability recognized. In addition, he serves as a scientific advisor, trustee, or patron for several autistic charities, and he has taken part in many television documentaries about autism. These remarkable scientific and public activities of Professor Baron-Cohen have been recognized recently in Britain's New Year's honor list, where he received a knighthood title. So without further ado, please welcome Sir Professor Simon Baron-Cohen.
Hi, thank you very much. Okay, great. So shall I try to make up for lost time? Can we go back one slide? Great. So I knew that Israeli security was the best in the world. I didn't realize I couldn't get my PowerPoint to show. But here we go. So thank you very much for inviting me to the conference. I wish that I could be there in person. And with Israel leading the world in terms of the vaccine program, I'm sure travel to Israel will be possible very soon. So I'm going to address this question about the relationship between autism and sex differences. Can I have the next slide? This is just to mention some of my collaborators in 3 different groups of types of research. The first are psychologists and imagers. The second are people looking at the role of hormones, particularly prenatal hormones and genetics. And the last group are collaborators in Denmark. I'll tell you more about their research when we get there. Next slide. So it's very sort of well known that there's a sex difference in the prevalence of autism. When I started my research career, people said it was about 4 males to every 1 female. You can see this data from the Center for Disease Control in the US, 2014, suggesting about 4 to 1. These days, people now say it's probably more like 3 to 1 or 2 to 1 because of, that females were just not being recognized and diagnosed for maybe many reasons. And as more females come forward for a diagnosis, the sex ratio is shifting, but there's still a male bias. Can I have the next slide? So this data from 2019 shows that when females do get their diagnosis, they're usually diagnosed later, suggesting that either they're not being referred at the same point that boys would be referred, maybe girls are better at hiding their diagnosis, or maybe concerns are not being raised as early, or maybe clinicians are not really very good at spotting autism in females. But nevertheless, females are now being picked up, and the sex ratio in diagnosed autism is still biased towards males. Next slide. So in my talk, what I want to do is explore some of the causal factors that might be influencing how autism manifests differently in males and females. And I think this is a kind of obvious approach, but sometimes it's important to spell out the obvious, that we need to look at potentially differences in genetics that differ between males and females in terms of what might influence their autism likelihood. We could also look at differences in prenatal sex steroids that might be, again, different in males and females in terms of affecting autism likelihood. And of course, both genes and prenatal sex steroids, hormones, are going to be impacting on prenatal brain development. So this is the kind of overall story that I'm going to be talking about. And the reason for focusing on these prenatal factors, genetics and sex steroid hormones, is that both of them have developmental programming effects on later development of the body, the brain, the mind, and behavior, and are sometimes said to have organizational effects, meaning long-term and maybe permanent. Next slide. So many of you will have seen this pie chart, which just shows what percentage of the variance in autism we can attribute to genetics and to non-genetic factors. And within the genetics, we can see rare genetic variants in the small slices of the pie chart, and then common genetic variants in that large red slice of the pie, and then non-genetic being the other unaccounted variants. So next slide. What we know is that many rare genetic variants have been identified. Many of you know that if you look at the Safari Gene website, you'll see a list of more than 100 rare genetic variants that are so-called high confidence genes for autism because they've been independently replicated in different labs. Next slide. These genes are usually expressed in the brain. No surprise, because autism is neurodevelopmental. But more interestingly, they're expressed prenatally in the brain at different points from early fetal to mid-fetal development. Next slide. And what we can see is that there are now some new studies suggesting sex differences in the frequency of some of these rare genetic variants. So looking at duplications and deletions that are de novo, you can see in several different studies that males and females with autism show different rates of these rare genetic variants. Next slide. What about common genetic variants? Well, the latest genome-wide association study is from Grove and colleagues in 2019, and this shows a Manhattan plot that many of you will be familiar with. So all of the chromosomes along the x-axis, and that red line going across the graph shows you the significance level that any of these common genetic variants or single nucleotide polymorphisms have to cross the red line in order for us to conclude that they show significant association with autism. So these are common genetic variants. We all carry them, but they occur in different versions. And you can see that in this very large genome-wide association study, 18,000 cases, 28,000 controls, they identified just 5 common genetic variants that crossed the line. This is telling us that you need big data to find common genetic variants. And if we want to see whether common genetic variants are contributing to sex differences in autism, we would need even larger samples. So this is work that needs to be done in the future. Next slide. But we know that autism is not just genetic, because you can have identical twins, like these sisters, where one is autistic and the other one isn't. And from some studies that have looked at so-called discordant identical twins, you can see differences in gene expression, so evidence of epigenetics at work, for example, changes in DNA methylation. And what I'm going to be arguing in this presentation is that one of the environmental factors that might be acting on the genetic predisposition to affect sex likelihood and sex presentation in autism are prenatal sex steroid hormones. Next slide. But let's start by just looking at the brain. Can we go back one? That was, yeah, thank you. Let's start by looking at the brain to see if we see evidence of both typical sex differences and their relationship to autism. First of all, in brain structure. Next slide. So this shows you some data from the UK Biobank, so published in 2018, looking at just over 5,000 typical brains, looking at white matter differences. And you can see there are typical sex differences in white matter on 2 different measures. Next slide. And looking at grey matter, you can see both total brain volume, grey matter volume, and white matter volume show typical sex differences in this sample of randomly selected individuals from the UK who have all donated both DNA and have had brain scans. Next slide. So some approaches have looked to see whether autistic people show an unusual association with typical sex differences. This was a study by Meng-Chuan Lai and colleagues back in 2013, which did a so-called spatial overlap, that if you first look in the brain at where are typical sex differences and plot those, and then look in the brain at differences between autism and controls, so a case control analysis, and plot those, and then do a spatial overlap. So what percentage of differences between cases and controls also differ between typical males and females? You see a very significant overlap in autistic females. This is using volumetric-based morphometry. So in red are the autistic females. And we can summarize this as saying both in grey matter and white matter, autistic females show a masculinized brain structure. Next slide. We can also look at typical sex differences across development. So many studies have done repeat brain scanning to look at whether there are typical sex differences. In the left graph, you can see this between ages of 7 and 17, and the blue line on the left-hand graph is males, the red one is females. So differences in total cerebral volume, for example. And on the right, you can see developmental, so prospective, repeated measures, MRI study of autism and controls, autism in red on the right-hand graph, controls in blue. And what we can see here is that the autistic brain, it's a different x-axis metric now, it's 12 months to 72 months, but still longitudinal. The autistic brain is larger at each time point, so growing faster and bigger than in typical development. But looking at this side by side, we can say that the autistic brain may be showing signs of being an extreme of the typical male. Next slide. And this larger brain seems to be the result of more neurons. So this data comes from post-mortem studies from Eric Korshen's lab in San Diego, showing that if you count the number of neurons, particularly in the frontal lobe, there are 65% more neurons in the autistic brain than in the typical brain. So the larger brain seems to correspond to more neurons. Next slide. And some other post-mortem data shows you in even more fine resolution that if you look at individual neurons, on the right, an autistic neuron, on the left, a typical neuron, we're not only seeing more neurons, but more connections between neurons, because each of those white dots that you can see along the length of the neuron is the point where the neuron forms dendritic spines with its neighbors. So there seem to be more connections between neurons as well in the autistic brain. And that little graph in the center suggests that this is due to reduced apoptosis in autism, because they've divided the samples of post-mortem brains into younger and older. So the dotted lines is the autism group, the solid line is the control group, and you can see less of a decline in the number of these dendritic spines in the autism group, suggesting there might be less pruning or less apoptosis of neural connections. Next slide. So let's bring this back to typical sex differences, because we know again from post-mortem studies that if you do the hard work of counting the number of neurons in males and females, this is across age now, but now their units are years, so from about 10 years old through to senior citizens, you can see that on average, males have more neurons, 22.8 billion, than females, who on average have 19.3 billion. So there's a sex difference in the typical brain in terms of number of neurons. And what we saw earlier was that in autism, there are even more neurons. Next slide. What about if we sort of switch from brain structure to brain function? So some studies are looking at resting state using functional MRI. Next slide. And these are two studies that have been published recently by Dorothea Flores and colleagues. And the top one, published in 2018, shows that during resting state, autistic people are showing a shift towards maleness relative to typical controls in the default mode network. So some evidence at the resting state functional activity of masculinization in the autistic brain. Next slide. What about when we use MRI, functional MRI with a task, so cognitive activation? Next slide. Well, I'm going to show you two different types of cognitive activation that we've used in our studies. One is an empathy task, so using the eyes test that many of you will be familiar with. If you present this to the participant whilst they're having a brain scan, and also outside the scanner, so you can look at performance. Many of you know this measure where you have to say which word best describes what the person in the photo is thinking or feeling. And here she's a bit dispirited or a bit sad. You can see that this produces a normal distribution in the general population. On the bottom left, you can see typical females score higher than typical males, but autistic males and females score below typical males, so showing an extreme of the typical male profile on this task. But in terms of fMRI, we see typical sex differences. Women show more activation in the left inferior frontal gyrus on this task, and autistic people show less activity in that same brain region, again suggesting autism may be an extreme of the typical male brain. Next slide. And a very different, one back, sorry. That's it. So a very different cognitive activation is this test of attention to detail, the embedded figures test. So we can see performance data here, the average number of seconds it takes for autistic people to find that cube hidden in the overall design. They're super quick, taking on average 32 seconds, compared to males, typical males who are faster than typical females, 46 seconds versus 66 seconds on average. And again, in terms of functional brain activity, when you're looking for the cube as quickly as you can in the overall design, I've lost the slides. Oh, it's still there. Excellent. Thank you. That typical males show less activity in the posterior parietal cortex whilst performing this task relative to typical females. And autistic people show even less activity than typical males whilst doing the task. So again, we could interpret this as the autistic brain showing an extreme of the typical male brain. Next slide. Let's move to psychological processes. Next slide. And again, looking at empathy, this study was published in 2018 in PNAS by David Greenberg, who's at Bar-Ilan University. A very large sample of 600,000 people using the empathy quotient. So it's self-report. How easy do you find it to read other people's emotions? And what you can see is that typical females score higher in terms of empathy than typical males. Next slide. And that when we included autistic people in this study, so you can see not only large typical sample, 600,000 people, but 36,000 autistic people took part. You've got the typical sex differences, and then the autistic sex differences are reduced and are shifted over to the left, again suggesting that autism may be an extreme of the typical male profile. Next slide. We also use the systemizing quotient. So this looks at how interested are you in systems of different kinds, whether they're mechanical systems, mathematical systems, musical systems, which basically involve patterns and rules. You can see that now the typical sex difference is with males on average scoring higher than females. Next slide. And when we added in autistic people, you can see that autistic people, both males and females, on average scored higher than typical males, who in turn scored higher than typical females. But again, fitting this pattern in the data of autism being an extreme of the typical male profile, again, using very big data. Next slide. We took advantage of having these, this was all an online study, obviously, of having these very large samples to also give all participants the AQ, the autism spectrum quotient. And what you can see is a typical sex difference in autistic traits. So typical males show more autistic traits than typical females on average. And of course, autistic people, who have a diagnosis, are showing even more autistic traits, but again, shifted over to the right, as if autism represents an extreme of the typical male profile. Next slide. So what we've done with data like this is to plot both empathy and systemizing in a model of two independent dimensions, empathy along the y-axis, systemizing along the x-axis, and dividing the whole population into what we call brain types. So in light blue are people whose empathy is higher than their systemizing, and in light pink we can see the opposite profile, people whose systemizing is higher than their empathy. And in that gray area in the middle, we call this type B for balanced, are people who show equivalent levels of empathy and systemizing. And then we have the two extreme brain types. In dark blue, an extreme of type E, people who empathize to a very high level, but who systemize just to an average or below average level. And then in dark red, people who are an extreme of type S, so they systemize to a very high level, but their empathy might just be average or below average. And basically, next slide, what we see is typical sex differences in these brain types. So this is that very large data set, 600,000 typical people. In yellow are females, in green are males, and then 36,000 autistic people in red and purple. And you can see that the yellow dots are kind of shifted up to the top left. The green dots are kind of more clustered in the middle, just eyeballing the data. And the red and purple are shifted more to the bottom right quadrant. Next slide. But if we actually do a count, the frequency of these different brain types in the population, what we see is that more females in the population show this type E profile, 40% of them, where their empathy is higher than their systemizing. More males show the type S profile, so their systemizing is higher than their empathy. And autistic males and females are disproportionately represented in type S or extreme type S, where they're systemizing, looking for patterns and rules in systems, is stronger than in the general population, and sometimes to an extreme. But again, we can interpret this data as autistic people showing an extreme of the typical male profile. Next slide. And this is just a different way of plotting that same result, which is using what we call the D score, which is the difference between your empathy and your systemizing, where you see typical sex differences and autistic people of both sexes being shifted over in a masculinized direction. Next slide. So all of this talk about sex differences in the mind could just be the result of culture, socialization, learning experience. But to get evidence that some of these sex differences might be partly innate or due to prenatal biology, we looked at newborn babies before there's been much opportunity for culture to shape brain differences or psychological differences. So these newborn babies were looking at a face or a geometric design, and they were assessed for how long they looked at the social stimulus versus the non-social stimulus. And what you can see is that at 24 hours old, the earliest we could assess them, more girls than boys in typical development looked longer at the face, and more boys than girls looked longer at the geometric design. Of course, there was a percentage of babies who looked equally long at both, but I'm just showing you the percentage of boys and girls who showed a bias towards either attention to the social world or attention to the non-social world. And the fact that we see any sex difference at birth in terms of attention suggests that whatever the role of culture, and of course we know that it's significant, there must also be prenatal biological factors influencing what we find worth attending to, or what we spontaneously attend to in our environment. Next slide. If we relate this back to autism, again, looking at data from San Diego, Karen Pierce's lab, we can see that at the earliest age that you can diagnose autism, around 2 years old, if you just look at children who spend more than 70% of their time looking at the geometric design rather than the face, you can see that the probability that such children are autistic is 100%. It's quite a striking result, but seen in the context of the previous slide, we can see that this profile matches the extreme of the typical male profile in terms of attention to the social and non-social world. And I'm sure you'll hear more about this in Amy Klin's talk tomorrow. Next slide. So let's move to the final part of my talk, which is the role of prenatal sex steroids. Next slide. So I want to focus particularly on this pathway called the delta-4 sex steroid pathway. So it starts with progesterone and goes through different steps, 17-alpha-hydroxyprogesterone, then androstenedione, through to testosterone. So each of these androgens they're converted into the next androgen in the pathway through the effect of different enzymes. And all of them are known to have masculinizing effects on brain development prenatally. Testosterone itself is then converted into estrogen through the action of an enzyme called aromatase. And if we go back to the model that Norman Geschwind put forward in 1985, a neurologist working in Boston, he argued that these hormones have organizational effects on brain development. He didn't have the opportunity to study it in humans because amniocentesis hadn't yet been widely used. We've been doing that for the last 20 years. So what you can see here is the fetus surrounded by the amniotic fluid in the amniotic sac. And as you know, some women opt to have amniocentesis as a clinical procedure during pregnancy. And we've been asking such women if we can analyze the fluid for these hormones, particularly testosterone, but all 4 of these hormones in the sex steroid, the delta-4 sex steroid pathway. Next slide. When their babies were old enough, so following them up postnatally, we looked to see if there were any correlations between levels of prenatal testosterone, for example, and volume of different gray matter brain regions. And you can see lots of correlations, particularly in regions of the brain to do with language, like the planum temporale, or social cognition, such as the right temporal parietal junction. So this is showing in humans for the first time that these prenatal hormones do influence brain structure. We've known it from animal research for over 50 years, but this was the first demonstration in humans using MRI relating volumetric-based morphometry back to prenatal testosterone. Next slide. And in terms of relevance to autism, we were able, in our small-scale studies, to ask parents to complete the AQ, so a measure of autistic traits, when their child was 4 years old, but where we had fetal testosterone measures from pregnancy. So these are not autistic kids. These kids are only in the study because their mother had an amniocentesis. And you can see a positive correlation between prenatal testosterone and the number of autistic traits the child is showing 4 years later. Next slide. And when these children were 8 years old, we gave them an empathy test. So this is the child version of the eyes test. Which word best describes what the person in the photo is thinking or feeling? Here he's interested in something, but what we see is a negative correlation. So the higher the child's prenatal testosterone, the more difficulty they're having with empathy or social perception some 8 years later. Next slide. We also see relationship between the hormone prenatal testosterone and attention to detail on the embedded figures test, a task that autistic kids do particularly well at. But remember, these are just typical kids where we're looking at individual differences in performance as a function or in association with prenatal testosterone. And you can see the higher the child's prenatal testosterone, the better they are performing in finding the little triangle hidden in the overall design at 8 years old. Next slide. But to really test whether prenatal sex steroid hormones like testosterone and estrogen are having any association with diagnosed autism, we collaborated with the Biobank in Copenhagen because they've been collecting amniotic fluid since the 1990s or even earlier. So they had almost 20,000 samples of amniotic fluid in their deep freeze. They also have a register for who gets a diagnosis, the National Psychiatric Register, so we know who is autistic and who is not. So we compared over 100 autistic people to over 200 typical people in terms of their prenatal sex steroid levels. Next slide. And what you can see is that looking at the delta-4 pathway, so testosterone, but also all 4 of those androgens, autistic people were showing elevated levels of these prenatal delta-4 sex steroid hormones relative to typical males, who in turn were producing more of them relative to typical females. We know that there's a sex difference in the typical population because some of these hormones come from the testes, some come from the adrenal glands, some come from other sources like the mother and the placenta. But you can see that in the case of autism, there were elevated levels. So next slide. And then in recent studies using the same cohort, we were able to look at prenatal estrogens, such as estradiol, again finding that the autistic group showed elevated exposure to prenatal estrogens compared to controls. Next slide. And bringing all this data together across all of these different prenatal sex steroid hormones, you can see that the odds ratios were particularly significant for estradiol, estrone, progesterone, and 17-alpha hydroxyprogesterone. They didn't reach significance in the later study for the other sex steroids, probably because we had reduced sample size. Next slide. This cartoon shows how the prenatal sex steroids are thought to operate. So what's the mechanism of action? So if we look at testosterone, it can cross the cell wall into the cytoplasm, where, as I mentioned earlier, it gets converted into estrogen under the action of aromatase. And once it's in the form of estrogen, it can bind to estrogen receptors and enter the nucleus of the cell to act as a transcription factor. And as I mentioned earlier, it's thought that the hormone binding to the receptor can change gene expression and in turn affect the rate of apoptosis or selective cell death. Next slide. What's the evidence for this model? Well, we can look both in animals and in human model systems like induced pluripotent stem cells and organoids. I'll come on to that in the next slide. But just in animal models, this classic study from 1976 shows that if you look at female rats and look at the sexually dimorphic nucleus of the preoptic area, on the left is what a typical female rat's brain region looks like. And on the right is what a typical male one looks like. Many more neurons and many more connections between neurons. But what we're looking at here is that when you take a typical female rat and give her extra estradiol, so one of these masculinizing prenatal sex steroid hormones, her brain is masculinized to resemble the typical male rat. So the hormone is changing brain structure prenatally. Next slide. And this data can be found on bioarchives, not yet published, but it's by Madeline Lancaster's group here in Cambridge using organoids or mini brains. So where you start with the 2D induced pluripotent stem cell derived neurons. So you're growing a brain in a dish. You let it grow into a 3D model, so it's so-called organoid, and then you add a potent androgen like DHT, and what you can see is it results in an increased number of excitatory neural precursors under the influence of extra androgens. So again, a demonstration that the prenatal sex steroids are changing brain structure. Next slide. I'm coming to the end of my talk, but this shows you an animal model from a group in China showing that when you use a drug called letrozole in the rat, which blocks the conversion of testosterone to estrogen, so you end up with excess testosterone, you can see that the treated animal shows reduced interest in spending time with a conspecific, a model of another rat, relative to spending time in an empty cage. So elevated prenatal testosterone in an animal model is results in reduced social interest in the offspring. Next slide. Could the elevated prenatal sex steroids be coming not from the fetus, which is possible, but from the mother? Well, in this study, again from that Chinese group, they looked at testosterone levels in mothers of autistic kids, that's the left-hand scatter plot, and typical kids, that's the right-hand scatter plot, just in serum samples, and finding that mothers of autistic kids have higher levels of testosterone, suggesting that the mother could be the source of the elevated testosterone that we were picking up in amniotic fluid. But of course, it could be coming from several sources. Next slide. And just as a clue that mothers may be contributing to the elevated sex steroid hormones in autism, you can look at a very specific subgroup, a medical condition called polycystic ovary syndrome, or PCOS, which is characterized by irregular menstrual cycles and late onset of puberty and excess bodily hair, or hirsutism. And if you look at these women who have this condition, which is caused by elevated testosterone, they have a higher likelihood of having an autistic child. This has been demonstrated in 4 different countries, including the UK by our group, but also in Israel and in Sweden and Finland. Relative to controls, the odds ratio is above 1 in all 4 of these independent cohorts, suggesting that women who have elevated testosterone for medical reasons have a higher likelihood of having an autistic child. So the sex steroid hormones may be influencing likelihood of autism. Next slide. In fact, this is my conclusion slide, and I hope I'm finishing roughly on time, which is that what I've tried to do in this talk is bring together prenatal influences in both typical sex differences and autism. I started off by talking about genetics, because clearly that must be part of the explanation for autism and, in all likelihood, typical sex differences. So we can see that in the top left. But lower in this graph, you can see the changing hormone levels. So you can see how prenatally we're looking at sort of changes across pregnancy in this schematic. But you can see the hormones, that the light blue rising curve is testosterone in males prenatally, is much higher than in females, shown in red. And if we look at estradiol, one of the estrogens, again, that rises really sharply during pregnancy. It is said that a woman's estrogen levels during pregnancy are higher than her estrogen levels in the rest of her life. So pregnant women have very high levels of estrogens. And as we've seen in some of the studies I've mentioned, both testosterone and estrogens have masculinizing effects on brain development. And the model here is showing that we have to look at genetics, prenatal sex steroids, including the hormones that come from the placenta itself, which can synthesize these hormones, and how both genetics and hormones change brain development across prenatal development. Last slide. I want to thank you for listening. Apologies for the late start, little technical issue. I want to thank our funders and invite you to visit our website for any more information about the studies I've been discussing. Thank you.
Thank you very much, Simon. I apologize for the technical issues that we had at the beginning, but I'm happy that we solved them eventually and were able to hear your wonderful and enlightening talk. It just means that next time we need to meet in person and hear your presentation again in person.
I was very happy to be able to come to Israel in June 2019 to Mount Scopus. And I was hoping to come, but then we had the pandemic. So, Lashana Haba.
Yes, we will surely invite you for the next meeting. We have a few questions from the audience. I'll start with my own question. So we see that there is a very strong correlation with exposure of testosterone and with a risk of autism. Is that means that maybe autism is reversible? Can we maybe use some hormone therapy to reverse autism, or maybe improve the severity?
Sure. So let me give you the scientific question, the scientific answer first, and then we'll have maybe the ethical answer. Can you hear me okay?
Yes.
Yeah, so the scientific answer is yes, of course, you could, in theory, you could use a testosterone blocker to reduce the amount of testosterone that the baby is exposed to. Should you do it? Ethically, I don't think you should, for two reasons. One is that we know that testosterone and other prenatal sex steroids are not only involved or associated with autism, they're associated with a whole range of outcomes, including aspects of the mind, aspects of talent, potentially, and gender identity and sexual orientation, etc. So we know that these hormones have widespread impact across the body, the brain, the mind, and behavior. So we shouldn't really be experimentally manipulating them without realizing there'll be a huge cost to the individual in many different systems. And secondly, myself, I am not looking for treatments for autism. This is, again, an ethical position, because autism is not just a disease or a disorder. Autism does involve some disability, inevitably, but often involves strengths, even talents, and certainly difference. This is part of the neurodiversity model. So I think we should be offering support for individuals and for families, but not be trying to, let's say, prevent autism.
Wonderful. We have some more questions. I read them from the chat. So Dr. Gamir is asking, if a male autistic child improves over time, do you think that we will be able to see in imaging a change in his brain or more to a more feminine brain?
It's very interesting. So the evidence I was presenting suggests that certainly the hormone effects are prenatal, and I'm not aware of evidence that that there are brain changes related to sort of your where you are situated on this spectrum of let's say maleness to femaleness across postnatal development. But I think it's an empirical question. I'm not sure that anyone's done the studies. What you would need effectively is longitudinal data across development with measures of, let's say, social functioning to see whether there is indeed plasticity. Geschwin's view was that these are kind of permanent changes that happen in the brain, but it is an empirical question.
Thanks. Another question from Professor Ilan Dinstein. Could you please share your thoughts about how these findings may impact early interventions, perhaps even during infancy?
Sure. So I was mostly focusing in my talk on the causes, the prenatal biological causes of typical sex differences and their link to autism likelihood, that's the sex ratio, and manifestation, why autism may look different in males and females. I wasn't really looking at intervention, but I think if we do this kind of early prenatal research, these may give us biomarkers where we could identify autism earlier and use existing interventions. I know that Ofer Golan at Bar-Ilan University has recently completed a trial using ESDM, the Early Start Denver model. It's one method of early intervention. At UCLA, they're using another method of early intervention called JASPER, Joint Attention and Symbolic Play and Emotion Regulation. So there are existing early intervention treatment methods, but I think that the prenatal biology could be identifying which children we should start with these behavioral or psychological interventions and starting as early as possible, if that's what parents want.
Okay, so maybe one more question. So we have a very clinical audience. So a question from Idan Weitzman. What can we, as clinicians, do better to identify autism in young girls and not misdiagnose them?
So what can we be looking for in young girls?
Yes, how we can diagnose better girls with autism earlier.
It's a very important question. There have been a number of studies recently looking at autistic adults, showing that autistic women report more camouflaging or masking of their autistic symptoms, and they say that they were doing this even in childhood. So I think what we need is studies of autistic girls at the earliest age to find out what camouflaging or masking strategies they're using so that we can be better at spotting when they're doing this. So why are they doing this camouflaging? Why are they hiding their autism? That could be because autism still carries some stigma, and that the stigma has more impact on girls than boys. Maybe there are greater social pressures for girls to be sociable and for girls to be communicative and playing in groups. And so if you are an undiagnosed autistic female, maybe you're spending more effort at hiding your autism, we want to make it comfortable for females and males to be able to be themselves. No need to hide your autism, but equally we need clinicians to be sensitive to when girls may be using these masking strategies, such as imitation, so that we can explore in more detail, does this child need an autism assessment?
Okay, I believe we have time for one last question. Question about the correlation. So is there any correlation with the severity of autism and level of high prenatal sex hormone?
So in our big study with the Danish Biobank, we didn't have access to severity of autism. It was just binary, is this child autistic or not? But you saw in our smaller studies where we could look at autistic traits that there was indeed a positive correlation, that the higher your level of prenatal testosterone, the more autistic traits. So if you use autistic traits as a metric of severity, then it looks like there are some clues. The problem with these studies is they're difficult to do. Collecting samples of amniotic fluid is quite invasive during pregnancy. In many parts of the world, amniocentesis is being phased out because of newer techniques for prenatal screening, for example, for Down syndrome. I'm aware that in Israel, amniocentesis is still widely used. And so we're planning some new studies in collaboration with Sheba Hospital to try to replicate some of our studies to look at the relationship between prenatal sex steroids and autistic traits whilst there's still an opportunity, because these methods will probably vanish and we'll have missed the opportunity to look at the critical window in prenatal development to measure these hormones in an ethical way.
Okay, there are more questions, but I think it's enough for tonight. We're already a little bit late. So once again, I want to really thank you for your effort, and apologize for the technical issues, and really hope that we will see you next time in Israel in person. So thank you again. A wonderful talk. And I want to...
להתראות.
להתראות. And thanks again for all the participants and the organizer for this wonderful day. Tomorrow we will have another great day program. We will meet you tomorrow at 5 at the same place and the same time. And see you tomorrow.